in the second half. So,
And my
so
morning
we've
is
got,
ready.
pretty much you're getting the idea that we start at the top and go through the hypothalamus and the gonadotropin releasing hormone, which you remember is just one hormone for two pituitary hormones, follicle stimulating hormone and luteinizing hormone. And then they trigger activities that go on to develop testosterone and for the production of sperm. And so the one new hormone we're going to talk about today is inhibin, and then when we get to the female, we'll add another one called activin. And as usual, we're going to talk about the pharmacokinetics, a little bit about transport, biotransformation, what the active compounds are from the testosterone, and some of the life cycle activities that regulate the amount of testosterone. And then tomorrow, Dr. Eldridge is going to talk about the myriad of drugs that are involved in reproductive pharmacology. And it's more than just birth control pills. So starting at the top, our hypothalamus, we've got some feedback mechanisms here, but predominantly we have just one hormone, gonadotropin releasing hormone, that's going to be triggering release in the anterior pituitary of luteinizing hormone and FSH. And so you remember that follicle stimulating hormone and luteinizing hormone are large protein hormones. They're dimers, and in fact they're glycoproteins. And the alpha and the beta subunits are what form the dimer, and which one is identical? Hmm?
The alpha.
The alpha. Does everybody agree?
Yeah.
Tyler, with Tyler? Okay, this is truth or dare.
Yes.
Okay, everybody agrees with that? Okay. It's on your head. So, half-life about 30 minutes. It gets to, this is follicle stimulating hormone. So what are the follicles, so to speak, referring to? Uh-huh. Yes. But they're not really follicles. So in the male, we're not talking really about follicles, even though we call it follicle stimulating hormone. In males, we're talking about those cells that are the nurse cells that are going to be providing the ability to let the sperm grow. And that is exactly right, Sertoli cells. So now, luteinizing hormone. Is there anything luteinized in a male? No, but it's the same name anyway. So luteinizing hormone can also be called interstitial cell stimulating hormone, because whereas you have the seminiferous tubules where you're going to be developing those sperm, in the cells that are the interstitial cells in between is comparable to those cells that would be the luteinizing hormone cells in the females. So really we have two false names here when we're talking about males. We don't really we don't have a follicle, and we don't have anything to be luteinized, but it's the same hormone, so we just have to remember that. So sometimes this is referred to as ICSH, but you know what, not usually. For the most part, it's always referred to as luteinizing hormone. And this one lasts a lot longer. This is 13 hours, and its target is the Leydig cells. And the Leydig cells are the ones that are going to be making the testosterone. So then our testosterone analogs are made here in the testis, and testosterone, dihydrotestosterone, are the two that we're going to be mostly working with. But keep in mind there are several weaker androgenic hormones like dihydroepiandrosterone and androsterone itself. So testosterone is the main source here, but some of these other androgens are coming from the adrenal gland. So this is not the only source of androgen in the male or female. Okay, now our new hormone that we're going to be talking about is called inhibin, and this has two functions. It can also be called anti-mullerial Mullerian hormone. And, you know, these all have their little abbreviations, but that would be during development of the fetus, in which if there isn't a signal with the male in order to suppress all of the female parts, then the female parts are going to take precedent. So you need to have something that suppresses it, and it's this hormone here. So on the other hand, in the fully developed male, this is a feedback mechanism that is made by the Sertoli cells in order to feed back all the way here to the pituitary and the hypothalamus to indicate that the Sertoli cells have received enough FSH, and the feedback is to block the FSH. So that's our feedback level here. Okay, now let's talk about this, because this is a new hormone to us, and this is part of the TGF-beta family. So are any of you familiar with the concepts of TGF-beta? It may have been something you may have discussed in cell biology, or when you're talking about cancer regulation. No? Yes? Sort of? Okay. So, so this is a family of peptides that, that come as dimers, and in the inhibin family, there's always going to be an alpha and a beta. And that beta can be beta A or beta B, and they're connected by 2 disulfides. It's glycosylated on the alpha subunit, and these are really what you might call cytokines. So they are going to be feedback mechanisms here in the reproductive system, but the whole TGF-beta family is really a regulator of growth and suppression of growth. So, so that family is involved in many, many other things. Today we're just going to be talking about the inhibin, and then we'll be talking about activin in the female cycle. So when the Sertoli cell responds to FSH, the FSH begins to induce new inhibins, and they get into the general circulation, and they're a feedback mechanism to the gonadotrophs that are in the pituitary, so that it can decrease the FSH release. So this is just one of the mechanisms by which the gonadotrophin-releasing hormone decapeptide can actually cause regulation of both FSH and LH released from the same cells, is that the feedback mechanism can be there to regulate which one of these hormones, LH or FSH, is going to be made, and which is going to be released and how it's going to be released. So this is our feedback. Now these inhibins are not just found in the Sertoli cells to feed back through the bloodstream. They're also made in the pituitary, the brain, the adrenal, the kidney, and in bone marrow. So probably in bone marrow they're working as a cytokine. It's not clear to me what they're doing when they're released from the adrenal and the kidney. So, or actually the pituitary or the brain, but I'm assuming that pituitary and brain can work as a feedback to FSH as well, if that's the case. But my guess is that there's probably a lot more to inhibins than just what we're discussing right now with feedback mechanisms. Okay, so let's now take a look at what's happening in the testis and how we're responding to these hormones. So the testis is made up of seminiferous tubules, which is what we see here. And surrounding the seminiferous tubules, you see these Leydig cells. That's why they're also called interstitial cells, is they're really not part of this seminiferous tubule. So what you see here in the pink are the initial beginnings of a sperm called spermatogonium. And they begin on the perimeter, and then they are completely surrounded by these green cells, which are the Sertoli cells. And the little purple dots here are the nuclei of the Sertoli cells. So you can see that there are junctions here that will hold these Sertoli cells together in a very strong connection so that you don't really see anything passing through. And in fact, what you see here is the Sertoli cell sort of surrounding these sperms so that they're covered on both edges by Sertoli cells. So the Sertoli cells are there as nurse cells to support these as they mature. And as they mature, they move closer and closer to the interior of the tubule. And once they've matured, their little tails are sticking out here into the tubule. And the Sertoli cells are here so that they're providing the only nutrients that the sperm is ever going to get because they're completely surrounding the sperm. And they're taking away any toxins. They're providing the nurturing hormones. And by the time the sperm has developed, these Sertoli cells are also making the fluid that goes into the seminiferous tubules so that as the mature sperm are released into the tubule itself, the seminiferous fluid goes with it. And you may have remembered that we had several different kinds of glucose transporters, and I told you that one of them, I think it was GLUT5, was a fructose transporter, and it was found in sperm. So one of the sugars that's being released here is fructose. So sperm seem to run on fructose, and they've got the transporter to carry it into these little sperm after they've matured. Okay, so our Sertoli cells are making growth factors, and it's all under the regulation of FSH. This barrier system can also be called the blood-testis barrier. So it's not like a plastic bag wrapping up your endothelial cells in your blood supply. What it is your Sertoli cells totally surrounding and nurturing your sperm as they develop. So they're not going to be letting in anything that doesn't belong. So what does FSH do? Well, so here's another view. This is our seminiferous tubule. It doesn't have nearly the detail that we had just a minute ago. But these cells that are here are going to be the only cells that are going to have the FSH receptors. And they'll also be expressing androgen receptors so that they can respond not only to the FSH, but also respond to the androgens. And that's all important in developing those sperm. Then they're also going to be making that Mullerian inhibitory substance during fetal development so that as the fetus begins to develop the testis, it suppresses what is going to be uterus and cervix of what would be the developing female. So this is a suppression system, and this is under that influence of FSH as it begins to start the development of the testis. So we get testis development, at the same time we're releasing this Mullerian inhibitory substance in order to prevent female body parts from developing. Now, in the adult, we're developing inhibin, and inhibin is going to feed back to regulate the FSH release. Now, the other thing we're doing is, since we're going to be dealing with androgen, and we've got our androgen receptors, we need to have an androgen receptor binding protein in order to keep the testosterone here where it's necessary for the development of the sperm. So the FSH calls in the production of androgen binding protein. That's a local protein. Now that's not the protein that delivers the testosterone to the rest of the body. That's a testosterone binding globulin. And we'll talk about that in a few minutes. But this is an androgen binding protein, is what it's called. And it keeps the androgens here for sperm development. And then the other thing that it does is takes the testosterone that comes from the Leydig cells, and you see your Leydig cells are down here. These are the ones that have the little nuclei now in this diagram. And these are making your androgens. And as the androgens pass through to the Sertoli cells and are kept here with the androgen binding protein, they also encounter the aromatase, which is going to change them from testosterone into estradiol. And estradiol is important for sperm production as well. So our estradiol is made locally by the Sertoli cells from testosterone that was made from the Leydig cells. So you could call this a paracrine effect because the estrogen that's necessary for sperm production comes from these Sertoli cells. And I don't know whether Dr. Eldridge reminded you, this is aromatase is also called CYP19. Did you know that? No? Okay. Well, you can surprise Dr. Eldridge tomorrow and talk to him about CYP19 and see if his face brightens up. So let's skip over and now look at what our Leydig cells are doing. Our Leydig cells are predominantly important in making that testosterone that's going to be important for the Sertoli cells to help create the sperm. So, so here now our diagram shows us the seminiferous tubules as these little curved edges. And now we're looking at the interstitial space here. And these are our interstitial cells of Leydig. But we also have some blood vessels here, because this is where the blood supply is coming and going. So that's very convenient, because now the cholesterol, which is the first step in making testosterone, can come in from the blood supply. And the first thing it's going to encounter here is our interstitial cells. And also, if we're going to be releasing our testosterone that we've made into the blood supply, it's being released right here directly from these Leydig cells. So we've got cholesterol, we've got several little arrows here until we get to a weak androgen, androstenedione. And then we've got an important enzyme, 17-beta hydroxysteroid dehydrogenase, which converts this to our stronger androgen, the testosterone itself. So it's luteinizing hormone that directs everything to do with Leydig cells. So if you're getting confused about your memorization, FSH for Sertoli cells and Leydig cells for luteinizing hormone, and you can kind of keep that all straight in your head. So luteinizing hormone promotes the induction of two proteins that are very important in bringing cholesterol into the cells and keeping it there, and then that would be the sterol carrier protein. Sterol carrier protein is important in binding a lot of the cholesterol-like molecules, and so it's not absolutely specific for cholesterol itself. And then we need another transporter that will take it to the mitochondria, and that's called steroidogenic acute regulator, or STAR. So with those two made, then you can begin to initiate the pathways that are going to take you right to testosterone. And that's going to be taking place here in these Leydig cells, but it's not going to be taking place along all of these lines in the adrenal glands. So that's where your differences come. Now let's put it all together, because now we have a Leydig cell over on this side of our blood supply, and on this side we have a Sertoli cell. And you can see the way this looks is that we've got this little curved space here, so that indicates that we're part of our seminiferous tubule. Here we've got a Sertoli cell that goes from the basilar side all the way to the apical side, where your mature sperm are getting ready to leave. So this is where the lumen is over on this side. So on this side is your interstitial cell of Leydig, and it doesn't have anything on it except interstitial space. So we've got our luteinizing hormone receptor. That's a G protein coupled receptor that activates GS. So we get more cyclic AMP, and that activates protein kinase A. and that's one of the primary signals for new protein synthesis. So those enzymes that we just talked about that are important for carrying in cholesterol and making testosterone, those are some of the new enzymes that are made so that we can get cholesterol and testosterone. Now our testosterone is important to get into the bloodstream, to go to all of the rest of the organs in the body that are going to be targets for testosterone. But in addition, it can cross over in a paracrine way and regulate activities going on here in the Sertoli cell. So our testosterone is very important for making and nurturing the sperm. So we want to be able to keep it here. So here our FSH is stimulating. This is where the receptors are on the Sertoli cell. Again, those are GS coupled, and we get more cyclic AMP, protein kinase A is activated, new protein synthesis, and one of the enzymes that's being made is the androgen binding protein. So the androgen binding protein can bind up some of the testosterone and store it so that it's going to be available for continued sperm production. Some of the testosterone is going to be acted on by the aromatase, which is one of those new proteins synthesized in response to FSH. And so now we make estradiol. Well, this estradiol is lipid soluble. It can cross back over here, and it can get to the proteins that need to be made in the Leydig cell. So the Leydig cell can respond to estradiol. And it's the estradiol that was made by the Sertoli cells in the seminiferous tubules. So new protein synthesis can come from the transcription factors here as well. So our Sertoli cells are allowing our spermatogonium to mature into sperm, releasing the sperm. And in addition, some of our new protein synthesis is involving other kinds of growth factors. And again, we can have paracrine communication here between cells so that our growth factors can go back and maintain the Leydig cells. So we've got lots of interactions between these two cell types. Now let's take a look back at our feedback mechanisms. And this time we're going to add a little extra nuance here because in the central nervous system we have lots of triggers that are going to begin triggering the hypothalamus, not only for release of FSH and LH, but also various kinds of reproductive behaviors. The mammillary bodies are very important in some of the reproductive behaviors that are taking place in the hypothalamus. So all of this can get coordinated, and ultimately we get little spikes from a pulse generator that's coming into play in the hypothalamus that can dominate in releasing the pulsatile release throughout the day. So you can see over the time of day we have pulsatile release just like we do with any other anterior pituitary peptide. And you can see sometimes it's bigger than others. But this release is going to be telling us a little bit about how this is going to be regulating the release of either LH or FSH. Because if we get low frequency pulses and they're fairly weak, then what we end up releasing is mostly FSH. But if we get high frequency and high amplitude pulses, then that's what dominates in releasing LH. So you can see your hypothalamus can play a role in this because even though it's releasing the amount that it releases and the frequency with which it releases is going to help determine whether you're going to get more FSH or LH vesicles made and released. So starting with our FSH, if our FSH is released, that goes to the Sertoli cells. And the feedback mechanism here is mostly through making that inhibin that's going to feed back to the anterior pituitary to shut down the FSH release. So if FSH is not going to be released, then you're going to get a predominance of LH released. Now the Sertoli cells are making all of these things that are necessary for making sperm, and also estrogens. And then they send some factors back over to the lutein or the Leydig cells. So the Leydig cells are going to be under the direction of the LH, and they're going to be making androgens. So our androgens are going to be largely the feedback mechanism from LH to shut down LH production. So you can see that you've got two ways. If you've got plenty of sperm being made and your Sertoli cells are functioning well, then you get the feedback mechanism to curtail the amount of FSH being released. And you know if the Leydig cells are making adequate amounts of androgens, then your feedback is to curtail some of the LH release. Now our androgens are also feeding back to the hypothalamus to help regulate the way that the gonadotrophin releasing hormone is being pulsed. And in addition, it's feeding back to the brain to to regulate behaviors that are going to be associated with reproduction. So we've got lots of reasons to be sending androgens here, but interestingly enough, the receptors that are here in the hypothalamus to respond are not going to be androgen receptors. They're going to be estrogen receptors in this feedback mechanism. So there's going to be aromatase here and aromatase here that's going to be changing those androgens into estrogens so that the feedback mechanism is largely estrogen related. Interesting. Okay, so here's our production beginning with our mitochondrial cholesterol to pregnenolone, some other things. We get to the pink area here where we start with our androgens with dihydroepiandrosterone, and then this is converted to androstenediol. We have another pathway if we cross over from pregnenolone to progesterone and go down this way, then we get androstenedione, and that makes testosterone. So your androstenediol can be converted to testosterone, and ultimately can be in the circulation either as androsterone or testosterone. And the testosterone, when it gets to target organs for androgenization, which is mostly the male secondary sex characteristics, it gets converted to a more potent testosterone called dihydrotestosterone. So that's not taking place here in the Leydig cells. This conversion is taking place in the periphery. And it results from an enzyme called 5-alpha reductase. And that's found largely in those cells that are going to be responding for male sexual characteristics. So here's our testosterone. It's converted to dihydrotestosterone. So dihydro means that it's got two hydrogens here where it used to have a double bond. So that makes it dihydrotestosterone. This is going to be much more potent at the androgen receptors, so that they're going to be responding here, and that would make testosterone a prohormone, because the more potent hormone is not testosterone itself, it's the dihydrotestosterone. But we have another way in the periphery of dealing with testosterone, and that is that we've got aromatase in many organs of the body. We've got aromatase where I told you it was in the pituitary and the hypothalamus for feedback. We've got aromatase in the bones where the epiphyseal closure is going to be responding to high testosterone levels in puberty in order to stop the long bone growth. And that's going to be via estrogen receptors. So we've got the aromatase there. And then we've got a lot of aromatase in fat cells. So when you see males that are obese and you see that it looks like they're developing breasts They are because they've got a lot of aromatase converting their testosterone into circulating estrogens. So those are some of our sources. We've got masculinization in the brain and fetal development, and interestingly enough, that's due to estrogens once they're converted as well. So this is contributing to many of the effects that testosterone is going to have in the body. And here our conversion is to take this ketone here and make it a hydroxyl. And in doing so, since we had double bond here, now that actually moves that double bond over here, and now we have what is now a flat ring for estradiol, whereas we had a chair formation or something over here that had the ketone on it. So that's a very simple modification, but it really changes the receptors that are going to be responding to our testosterone. Now, once we get the testosterone in the bloodstream, it's transported by a sex hormone binding globulin, SHBG, and that's not the same as that androgen binding protein that was found in the males. So this is going to be taking up about 45 to 60% of our testosterone, and then the rest is going to be much less strongly bound to albumin. So we only have about 2% free, but it's that free form that's free to get across the blood supply and into the tissues. Now this binding globulin is made largely by the liver, and so it's subject to change. If you have liver disease, you're probably going to be making less of it, and you're also going to be making less albumin, so you have less carrying capacity. But on the other hand, this binding globulin can be induced, and it can be induced by estrogens, because it also binds estrogens. So since it carries both estrogens and testosterone, it's very important to have adequate estrogen levels during pregnancy when the estrogen levels are high. So estrogen will induce it. On the other hand, testosterone tends to reduce and suppress the amount of sex hormone binding globulin. Interestingly enough, this is also expressed in brain. It's not so unusual that it might be expressed in placenta or testis, but it's not real clear why it might be expressed in brain. But there's recent data that suggests that it might bind to a membrane receptor that can then help trigger some of the very short-term effects that testosterone might have. So while we think of it as a genomic receptor, it's a sex hormone, so it's going to be binding its transcription factors in the nucleus, but it has probably a mechanism whereby the sex hormone binding globulin can cooperate with some other membrane receptor for short-term signaling. Okay, how do we get rid of our androgens? Well, that's going to be taking place in the liver and in the kidney. And in the liver, testosterone is converted to androstenedione, which is the weaker of the androgens. And dihydrotestosterone can be converted to another weak androgen, 3-alpha androstenediol, and these will be converted to 17-ketosteroids. And those are much more water-soluble. So it's our water-soluble weak androgens that come out in the urine. And if that's not enough to make them water-soluble, then they're conjugated with glucuronide or sulfates in order to make them even more water-soluble so they can come out in the urine. So the kidney can excrete about 50% in terms of these 17-ketosteroids, and the others are coming out as the conjugated steroids. Now the main mechanism that we think androgens work at is inducing new proteins. And so the way this works is they're very lipid soluble, so they don't really need to have a transporter that brings them into the cell. But then they meet their receptor out in the cytosol. And for the most part, steroid hormone receptors are waiting with a chaperone protein, which was chaperoning them while they were folding from the Golgi apparatus. And so they're going to be binding to their heat shock protein, which is their chaperone protein. And once the androgen binds here, it's going to release that heat shock protein so that either testosterone or dihydrotestosterone is going to carry that transporter across the nuclear pore. And there it can interact with the binding element for it on the chromatin. So for the most part, it can bind to testosterone, but it binds to dihydrotestosterone 30 to 50 times more potently, and it's much longer lasting. So while you have your androgen receptors and they can bind both, in fact they can bind some of the weaker androgens as well, but the one that is the most likely to have the best effects is dihydrotestosterone. So testosterone's actions. Well, we already talked about this several ways. The testosterone is going to be produced as the fetus is developing its testis, and at that point it's going to be helping to develop all of the sex organs of the male. So that includes the penis, the scrotum, the prostate glands, the seminal vesicles, genital ducts, and goes all the way to supporting the descent of the testis during gestation. So this is all taking place during fetal development. And then it declines as we get closer to delivery. And then shortly after birth, there's a small release of testosterone into the bloodstream of the neonate, but it's really not clear why that happens. So I'm not even going to venture a guess. But from then on, we have very little happening right up until about the age of 6 or 8. And then we have what we call adrenarche, which is a release from the adrenal glands of some of the weak androgens that adrenal glands can make. So this is pre-pubertal, so that it's the beginnings of getting ready for puberty, but it's not really. So the adrenal glands are making a weak androgen, dihydroepiandrosterone, DHEA, and that can be released from the adrenal glands at an earlier age. It peaks actually into adulthood and then begins to decline. But its release is under the influence of ACTH. Now, I don't know whether Dr. Eldridge is going to be talking about this or not, but you can purchase this if you want it. You can go to general nutrition stores, and it's been on 60 Minutes. It's been promoted for husbands who are couch potatoes and their wives want to perk up their sex life, and they think that they can provide them some additional androgen, but it probably doesn't do any good. Because there aren't any real studies that have shown that it increases muscle strength, or that it really does anything with respect to lipid profiles, insulin, glucose, nitrogen balance, bone mineral density, lean body mass, BMI, or libido. On the other hand, people will self-report that they have an improved sense of well-being, whether they're males or females. So, you know, it could be that if you spend a lot of money and you take a lot of pills, that you're probably going to think you feel a lot better than what you probably do. So, you know, if it works, I suppose it works. But You know, males have plenty of testosterone already, and that's a stronger androgen. So, giving them a weak androgen probably isn't going to do anything, and you know, for that matter, it might even be competing at those androgen receptors. So, so this may not work so well as it appears for males. If women take it, on the other hand, they don't have those strong androgens in their circulation, and they're likely to begin to start seeing androgenic effects. And they're probably not going to like it, because that includes acne, baldness, hair on your face where you don't want it, voice deepening, and that actually might be irreversible. And then if there are hormone-dependent cancers, these weak androgens might do something to stimulate those. So it's probably not a good idea to use this exogenously, even though it's advertised. Okay, so we started out talking about this as part of the adrenarchy. So now moving on to puberty. Puberty, we don't really know what it is that starts puberty, but it's hypothesized that the suppression that we had from the steroids, the small weak steroids are going to be sensitive enough in the hypothalamus to suppress any kind of FSH or LH release. And here's an example of that. Pre-pubertally, we have these little tiny spikes of very low levels of LH, but at some point it's believed that the sensitivity to these steroids that suppress that gets released, and that's what initiates puberty. So then you begin to see that the height of these pulsatile spikes gets to be higher, and particularly in sleep. And so that's what you see as puberty. So initially you see that LH is being released initially, and a little bit of FSH. And then by the time 9 or 10 years old, we begin to see a little bit more FSH. And so during this time, the gonads are really developing so that they can begin to, as they increase in size, are going to begin to get the Leydig cells responding and increasing our testosterone. And as the testosterone levels increase, we begin to get to see more sperm production. We see the penis growing, pubertal hair beginning, and a large growth spurt in males. So all of this is taking place as those plasma testosterone levels begin to rise to adulthood. So, so again, it's believed that that whole thing got started as our feedback mechanisms seem to lose their sensitivity. And then we see that kind of growth in all of the male sex glands and the secondary sex tissue, and libido increases. So all of this, now to look at testosterone levels, which are the ones that are in red, years 10 or 11, 12 years old, and then it begins to rise to adult levels. This is not to scale because all the way up to 60, 75, we maintain these high levels of testosterone. And the blue is our sperm production. So our sperm production lags behind the testosterone because you needed the testosterone in order to make the sperm. So that continues right on into old age. Old age, they're telling us, is 80. Well, maybe they started old age at 70, but here you can see that there is a decline in testosterone, but it never declines a whole lot. That is to say, it hasn't ever dropped below about 50%. And our sperm production, you've got plenty of sperm, so elderly gentlemen can reproduce just as well as they could have when they were in adulthood. So what is our testosterone doing during adulthood? It has, during puberty, increased the size of the penis, scrotum, and testis, so it maintains that during adulthood. It maintains all of the secondary sex characteristics, so that's the more testosterone, the more likelihood of getting bald. And it maintains the larynx. Increases the thickness of the skin, increases muscle mass, increases bone growth so that you've got maintenance of your bone mineral density and calcium salt deposition, and increases red blood cells because it induces erythropoietin, which is the cytokine that allows the maturation of the red blood cells. And here's our baldness. So, so we've covered a lot. I'm going to switch over to the female hormones. But are there any questions that anybody wants to ask before we leave? There's one thing that I forgot to say that tell me about what happens when a person takes exogenous androgens for bodybuilding.
Isn't it, you mean like anabolic steroids kind of
Uh-huh.
thing? Isn't there shrinkage of the testes?
Uh-huh. Now, why do you suppose that is, based on what we just talked about? It's, well, you've got feedback, and it is negative. And yes, so that's one aspect. So what happens then?
Natural production of testosterone.
That's right. So you feedback, now you have less luteinizing hormone. You're making less androgens in your Leydig cells. It's the Leydig cells androgen that is populating the Sertoli cell hormone receptors. So even though you may have a circulation of androgens, that's not where the source of androgens are for sperm development. That source has come directly from the Leydig cells. So if you are suppressing your luteinizing hormone release and you're suppressing your Leydig cell production of testosterone, then your Sertoli cells are not getting it. And they're not getting it to convert it into the estrogens either. So now you don't have your estrogens, you don't have your androgens, you've suppressed your LH, you've suppressed your FSH, and so bodybuilders are noted for infertility because their sperm levels have plummeted.
Bodybuilders that supplement or all bodybuilders?
No, supplemental,
Okay.
you know, people that are
Stop working out right now,
that are
right?
really- yes.
Is doesn't working out increase testosterone? Or no?
I don't know, but I do know that it suppresses. So you remember we talked about leptin. So leptin is released the more fat cells you've got, and leptin is a signal for advancing gonadotrophin release. So typically, people who are working out a lot and don't have any fat cells, so that they don't have that feedback by leptin, are not going to be getting the gonadotrophin release as well. So this has happened to me several times when I have a Zumba class instructor that I really like, or kickboxing or something, and then she has to quit because she wants to get pregnant and she's having trouble getting pregnant, and her doctor tells her to stop working out 3 times a day. And so, as soon as you begin to get enough of a supply of leptin being released that your brain realizes that you're in a good state to be able to reproduce, then you will. So I don't know what happens with men on that, but it could be perhaps the same thing. That if you're working out and the leptin feedback in males may be the same, that if you don't have a fat cell to speak of, then you might not be so able to reproduce. Okay, but on the other hand, don't count on that as a birth control. Don't say, Dr.
comment.
Howlett told me that if I worked out, then I wouldn't get pregnant.
Yeah.
Same thing, except now we're a little bit more complex. So we've got all of our same hormones. Now we're going to add another one of those TGF-beta-like hormones. This one's called activin, and we'll talk about that when we get there. But, so now we've got, here's some of our regulation we just talked about. How convenient, it's all right up here. So our positive stimulators include norepinephrine, NPY, and leptin. So these, norepinephrine is going to be a trigger of metabolism. NPY and leptin are the ones that are involved in feeding behaviors and the response to knowledge that you've got plenty of adipocytes. So you've got plenty of adipocytes, you've got this positive regulation here, releasing our gonadotrophins. On the other hand, the kinds of neurotransmitters and peptides that can block this include GABA, dopamine, interleukins, and beta endorphins. So let's think about this for a minute. So GABA is an inhibitory neurotransmitter. So maybe there's some inhibitory circuits that are here coming into the hypothalamus. Dopamine, where did we see dopamine as a regulator of pituitary hormones before? Prolactin and oxytocin. So go back and look at that diagram, because there's one little arm of that diagram that feeds back and negatively regulates FSH and LH. Now, don't think of that as a way of birth control either. It maybe suppresses the probability of releasing LH and FSH, but I wouldn't count on that if you really don't want to have a second child until a couple of years later. Don't count on breastfeeding to suppress it forever. Beta endorphins. So beta endorphins are peptide hormones. They're the ones that are made by cleavage of the POMC, pro-opiomelanocortin peptide. And so they are there as suppressors. And typically you've probably heard that people who are heroin or other opioid addicts may not have good sperm production either. And so here we've got a situation where that's allowing the suppression of LH and FSH release from the gonadotrophin releasing hormone signal. And then interleukin-1, so that's a pro-inflammatory cytokine. So if you're sick, you don't want to reproduce either. So that may be a way of helping to protect from reproduction in a chronically inflamed person. So these are some of our regulators. Our LH and the FSH are feeding down to the development of a follicle. And as the follicle develops, the cells that make up that follicle that are surrounding this little ovum here are going to be the ones that are making the estrogens and the progesterone. Well, they do it in a cyclic fashion. So this is not all taking place at exactly the same time, because usually we get our primordial follicles that are beginning to develop. And during fetal development, the female fetus has virtually all of the putative ova that she's ever going to have. And then every single month, 4 or 5 of these begin to develop. And then one of them gets to be larger than the others, and it suppresses the others, and they go through atresia. So you end up with one of them that takes over and begins to develop. And so here's the ovum itself, and surrounding it are fecal cells, and these are going to be making progesterone. And then you've got granulosa cells that are going to be making the estrogens. So, and again, that's 2 cell types that you need in order to make these estrogens. Then as that develops in the first 2 weeks of the cycle, we ultimately get what would be a mature ovum. When it's ready to be released, it suddenly gets a large bolus of luteinizing hormone that allows the release. And then in the second half of the cycle, these fecal cells are what we call luteinized by the luteinizing hormone. they begin to start making a lot of progesterone, and they're still making reasonably high concentrations of estrogen. So they begin to look a little bit yellow with all of this lipid material that they're making. So this is called corpus luteum, typically as yellow. And if you don't get a pregnancy, then it begins to degenerate and goes through atresia. And then it happens all over again. So this is the cycle. We're going to go through the cycle from the point of view of how the hormones are regulated during the different kinds of cycle, and parts of the cycle, and where the feedback mechanisms are. So we've got three different estrogens: estradiol, estrone, and estriol that we'll talk about. The primary, most potent one is estradiol. Estrone is largely coming from fat cells that converted it from androgens, weak androgens, and estriol is largely coming from the placental fetal unit, and progesterone. So let's talk about our estrogens first. Here's our estradiol. It's got an aromatic ring here and hydroxyl. And our estrone can be converted without the benefit of having to have an enzyme. So it can go from estradiol, a strong estrogen, to estrone, a relatively weaker estrogen. The estrone mostly comes from the androgens that are made in the adrenal, and then the aromatase that can convert them into estrone is largely found in adipose tissue. So women who have more adipose tissue are more likely to make more estrone. And this can be very useful if your ovaries are no longer making estradiol, because now this estrone can be converted to estradiol. So women who have more adiposity in menopause tend to have fewer symptoms of loss of their estrogens, because they have a source of it from the aromatase that can convert it from the adrenals. Then our estriol is most prevalent during pregnancy, and we'll talk about how that happens as well. Okay, so here's a cartoon of our ovum in the middle, surrounded by our granulosa cells and our thecal cells, and that's our follicle as it develops. So it's our thecal cells that are going to be making our androgens. They'll make androstenedione, dihydroepiandrosterone, and testosterone. And then these cross the barrier over to the granulosa cells, where the granulosa cells have the aromatase and can make the estradiol. So again, you've got two cells that are necessary for making these steroids. And the reason is that some have the enzymes that can do it and others cannot. So here's our thecal cells, our granulosa cells. They both can bring in cholesterol, so they both have those enzymes, just like we talked about in the males. And then the granulosa cells have to stop there at progesterone. They can't go any further along this pathway, but the thecal cells can now take it to 17-alpha hydroxyprogesterone, onto androstenedione, a weak androgen, and onto testosterone. But the testosterone does not, in abundance, get released into the blood supply here. Instead, it crosses to the granulosa cell, where aromatase immediately makes it into estradiol. And this androstenedione can be converted to estrone, which can be made into estradiol. So our granulosa cells are making a lot of estrogens, and particularly estradiol, because you can get there from all three of these mechanisms from the thecal cells. Now these are transported in the blood supply by that same sex hormone binding globulin, and again, most of it is bound to the sex hormone binding globulin, some is bound to albumin, and about 2% is free. Now interestingly enough, the progesterone is mostly bound to albumin with a little bit of gamma globulin. It does not bind to the sex hormone binding globulin. And as for the other steroids we've talked about, they're converted to glucuronides or removed in the urine. Now, we talk about our sex hormone binding, our sex hormones as being genomic receptors, because traditionally we've always thought that that they can bind to the receptors that are going to have response elements for estrogen, or estrogen receptor response elements on the promoter regions. And once they do that, then they call in some additional proteins that are going to be binding, so that ultimately we can initiate the polymerase II and get new gene expression. Now that's the way we've always thought it works, and we've got two receptors that can do this, the alpha receptor and the beta receptor. And they're going to be in different tissues of the body. So when you think about the tissues that are going to be responding to estrogen, you tend to think about the female sex organs, and they're going to have estrogen receptor alpha and beta. But you don't tend to think about too many other things. But typically our estrogen receptors alpha and beta are going to be in bone, and you remember they were going to be suppressing our osteoclasts so that you could get stronger bone growth. But you can see that our estrogen receptor alpha is present in the liver. So the induction of liver proteins by the estrogens is really very important in maintaining a lot of functions in the body. Here's our breast tissue, alpha and beta receptors. We've got alpha receptor beta in the lungs, and we've got estrogen receptors in the cardiovascular system, and plenty of alpha and beta receptors in the brain. So our genomic mechanism is working all over the body. But we also now have a receptor that we know of as being a G protein coupled receptor. And this is taking a good deal of what would be our very short-term actions. You know, if it's going to be a genomic response, it's going to take time in order for transcription to take place. It's going to take time for translation to take place. It's going to take time for enzymes to be modified and transported to where they need to be in the cell. So this could take a minimum of 30 minutes, maybe 2 hours. And if you're going to be accumulating things in vesicles, that's going to take even longer. So there are some things that estrogen does that are very rapid. And the question about how that works has always been a question until we got these orphan receptors from the Human Genome Project and discovered that the one that was number 30, GPR30, is now what we call the G protein estrogen receptor, or GPER, as the case may be. So GPERs are found in membranes, but not necessarily just the plasma membrane. So here we see a lot of GPER that's found here in the endoplasmic reticulum. And while this can be transported to the plasma membrane, where it can be responding to estradiol as a G protein coupled receptor, it can also be active while it's inside the cell, because estrogen can get inside the cell. So, so here it's going to be active as well. So it has several mechanisms. It's going through the PI3 kinase, MAP kinase pathway, and then that can interact in the transcription, not looking through estrogen receptors, but perhaps through CREB. And other effects that are going to be taking place that can change ion channel activities and other things in the cell. So I bring this to your attention because in the next few years, probably this is the primary target that drug companies can go after for selectivity. Are they targeting events that are short-term by estrogen, long-term by estrogen? How can we regulate one versus another? Very exciting, probably could be very useful for cancer chemotherapies and for postmenopausal regulation, where you don't want to have uterine growth or breast growth, but you want to have strong bone mineral density. You want to be able to regulate those receptors that are in the brain, so you want to be able to work with more Okay, so let's call our attention to regulation now. In childhood, we begin to see, again, what would be, well, let's go fetal development. So you get, as the ovaries are beginning to develop, you begin to see gonadotrophins involved in regulating the ovarian development here. So you've got FSH in red and LH here in yellow. And then a little bit of release in infancy for reasons that it's really not clear, and then this long period of time in which you don't see very much. Then it's believed, again, this sensitivity theory that in childhood you're very sensitive to feedback, but as adolescence begins to happen, then the amount of sex steroid feedback that can suppress LH and FSH is reduced, and you begin to see more LH and FSH being released, and then much less sensitive back in adulthood. So ultimately, we see that we can begin at age 10 to 14 in females where we begin to see LH and FSH being released. And then our main pulsatile peaks in LH are going to be occurring in those monthly surges that releases that ovum. So these are monthly, not minutes. And then as we get to old age, then we don't have that feedback anymore because we won't have the estrogens being made. So there are two ways that we talk about the menstrual cycle. We can talk about it from the point of view of the ovary, or we can talk about it from the point of view of the uterus. So from the point of view of the ovary, that first half is called the follicular phase because the follicle that contains the ovum is developing. And then once the follicle has burst open and the ovum has been released, we go into what is now called the luteal phase, and that's where luteinizing hormone is allowing those thecal and follicular cells to now make a lot of hormone. And it's mostly going to be the thecal cells, so you're going to see a lot of progesterone being made here. So 2 halves of the cycle, roughly. And right here at 14 days is where the cycle half begins, because that's where ovulation has taken place. Now, from the point of view of the endometrium, we start out with the first day of the menstrual cycle, and that's where the tissue is sloughing off of the uterus because there wasn't a pregnancy and it wasn't necessary anymore. And that's only going to take place for 4 or 5 days, and after that, the follicle has developed, it's making estrogens, and the estrogens are mitogenic. So from the point of view of the endometrium, this is where that tissue is building up again. So the endometrial cells and the myometrial cells are all undergoing rapid mitosis. So this first half of the cycle now has what the endometrium considers to be the proliferative phase. Then as soon as the ovum has been released, and now you're beginning to get a lot of progesterone that stops the estrogen's mitotic effects and now moves us into what's called the secretory phase, because now the cells are beginning to make glycogen, they're beginning to make other things that are going to allow a good implantation of that fertilized ovum, hopefully from the point of view of the endometrium. That's the endometrium's goals. So you'll hear both of these nomenclature. As we go through this, and we already pretty much did this, our cells are developing right up until our release of the ovum during our first half of the cycle, and then during the second half of the cycle, we get the luteinization, or the corpus luteum, which no longer has the ovum in it, and it's now making a lot of progesterone. And then if we don't have oxidation, then that goes into atresia and disintegrates. Okay, so let's go over our levels of hormones during these two phases, and we're going to start with our follicular phase. During this time, we've got FSH being released, and this is the first half of the cycle, so we're only going to go up to day 14 in this diagram. So the triangles here are FSH, and as the cycle begins, we get our peak of FSH around day 6 or 7, and then it begins to decline. And the reason it's declining is that inhibin levels are being made by those follicular cells, and they're going to be releasing that inhibin in much the same way that we saw the Sertoli cells releasing inhibin. And the more inhibin you get released, the suppression of that FSH. So that's just like it was in the male. So that's not terribly unusual for us. Now, as that follicle develops, it begins making more and more estrogens. So during this time, our estrogen levels are beginning to rise. So you can see it's after the FSH peak that we begin to see estrogen. You can see it's going up precipitously high here on day 14 or 13 or somewhere in the middle of the cycle. Now keep in mind your LH has been very low levels during this entire time, and your progesterone, which is the squares, are also at very low levels during the entire time. So that's the whole first half of the cycle, limited LH, limited progesterone. Now once the estrogen has begun to come into the circulation, we begin to see that our endometrial tissue is proliferating. So our uterus increases the number of cells in both the myometrium and the endometrium. Those cells are beginning to get oxytocin receptors in the, so that they can contract, and you're beginning to get a much stronger blood supply. In menstruation, that blood supply had been released. It had been reduced. It all got sloughed away. So now you're rebuilding that. At the level of the cervix, you've got increased dilation because you want to be able to get the sperm to come across. And so you don't want to have viscous fluid there. You want to see mostly saline. And when you put saline on a microscope slide, it crystallizes in a form that looks like ferns, and they call that a ferning pattern. That's to distinguish it from mucus, which is thick and viscous. So in the first half of the cycle, you don't want a viscous cervical fluid. You want to be able to get the sperm to come across. And what is typically used as a test to be able to see where you are in the cycle would be to take some of that cervical fluid. If it has the ferning pattern on a microscope slide, then you know you're in the first half of the cycle. If it looks messy and mucousy, then you know you're in the second half. So this is called ferning, and its German name is Spinnbarkeit, and is a typical diagnostic that's been used by women and physicians for eons of time. And you can do that without having to have a fancy radioimmunoassay. So at the level of the vagina, again, proliferation, oviduct, increased fluid secretion, and again, that's mostly saline. Now you want to get ready for pregnancy, so the body fat deposition is going to be under the influence of this estrogen, so that you can have plenty of fat available so that you could carry a fetus for 9 months and not lose energy. Here's where we've got some of those short-term effects that estrogen will be having, and that's increased sodium and water retention in those cells. And that's not going to require genomic receptors. That's probably under the influence of those short-term receptors. Then we've thrown in a couple of things here. The skeletal closure of the epiphyseal plates is under the influence of estrogen. So in males, that's where the testosterone was converted by aromatase to estrogen. So this is going to be taking place at toward the end of puberty, when you have a large amount of estrogens that are being released. Okay, now what causes ovulation? So now we've gone to beyond the 14 days here. Now we're up to this point here. And as our ovum has developed and our follicular cells are making and releasing lots of estrogens, those estrogens right here are getting into the circulation. And for 2 or 3 days, they're going to be at very high levels. And the hypothalamus is going to recognize that. So part of that is going to feed back, and along with the inhibin. Now the inhibin has been suppressing FSH all along, but now you see a big spike of inhibin here. And you see this high level of estradiol, and that's going to be suppressing the FSH release. But it's going to be getting ready to have a large amount of LH release. So the LH is going to start accumulating in these pituitary cells, and after 2 or 3 days of high estrogen levels, then suddenly what you see is a large spike of gonadotrophin releasing hormone, and that's going to be mostly releasing LH, which is here in the squares. So for the entire first half of the cycle, low, 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 and then suddenly, zap, you get a large amount of LH being released, and that's what gives you the ovulation surge.
How low?
Low, 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 low. Okay. Now over here, this is your depiction of your ovulation taking place right here at this exact same minute. And actually it's not a minute, it's about 20 minutes. It doesn't take very long for that whole process, that gonadotrophin surge, the LH surge, the ovulatory release, over and done with in 15 or 20 minutes. And then the ovum is free to find its way. You've still got that, you don't have mucus yet in your fluids, so you've got plenty of opportunity for the ovum to find its way down the oviduct as the sperm are coming up the other direction, and so hopefully they're going to meet right there. So at this point, we have this large surge, and you'll also note that the FSH has surged too, but the units here are in the tens of units per liter. Over here, the units are in the ones of units here. So even though it looks like they're coming out at the same concentration, they're not. So pay attention, the units are different over here. Okay, now at that point, once ovulation has taken place, you don't see quite as much estrogen being made anymore. And on the other hand, you begin to see now your progesterone is being made, because this surge of luteinizing hormone is going to now promote the corpus luteum to now develop in what we call luteinization, so that it can begin to make a lot of progesterone. And you see that here at our endometrial tissue, the proliferation phase has ended. Right here, this is ovulation. And now as our luteal phase here, you can see no more proliferation. But you begin to see what they call corkscrew production of what would be secretory patterns here in your histology. So that's where you're going to be releasing fluids into the tissue and promoting the opportunity for the fertilized ovum to embed. So now you have to go through what's now a differentiation, stopping estrogen's effects on proliferation. Now, you may get confused, because when I say negative feedback or positive feedback, you can say, well, why can you be negative and positive at the same time? Well, you're looking at different activities. So your estrogens and your inhibins are blocking your production of, or your release of FSH, and that's an inhibitory feedback. But you have a positive feedback of this 2 or 3 days in order to get this very large peak of gonadotrophin releasing hormone. And so that is a positive feedback. So your estrogen is going to have both negative feedback to suppress FSH and positive feedback to get ready to have that LH surge. And so the responsiveness at the pituitary is also going to be based on estrogen. So during the early follicular phase, when you don't have very much estrogen, you could have a little gonadotrophin releasing hormone pulse, and it's going to produce a little itty-bitty minor luteinizing hormone release. But late in the follicular phase, when you have plenty of estrogen, you can get largely much larger increases, and suddenly you get that very large increase in luteinizing hormone. Now the new hormone is called activins, and the activins are going to be stimulating FSH release. And so here you see that unlike the inhibins, the activins are homodimers, and again they're dimers, but now they are either AA beta, BB beta, or AB beta, and again they're connected by two subhydral links. So whereas the inhibins are released from the granulosa cells and can suppress FSH, the activins are made by these very same tissues, and they stimulate FSH. So beginning in the middle of that second half of the cycle, you begin to see activins being increased. So our luteinizing phase is our second half. So now we're looking, this is 2 weeks into the second half. We've just had our ovulation. This is our inhibin A, or our activin, and that's beginning to rise. So by the second week of the second half of the cycle, you're beginning to get this stimulation here. So that's building up the FSH levels so that you could go into another half of the cycle. But this is our activins being released. Now what's happening here? So our FSH has been suppressed, and that's our triangles. Our luteinizing hormone had that very large peak, but now it's come down. This little line here is coming down, and it's there, but it's not going to be quite as large, but it's enough to maintain the corpus luteum for several weeks. Meanwhile, our corpus luteum is going to be making plenty of progesterone, because it's mostly those thecal cells that have been luteinized. They're making a lot of progesterone, but you still have an increasing amount of estradiol being made as well. So we're going right up here almost until the end of the second half, because the assumption now is that we do not have an implantation, so we do not have any stimulus to maintain luteinizing hormone from the placental tissue. And so as the luteinizing hormone decreases here, you can't maintain that corpus luteum anymore. And so it can no longer make progesterone. So the progesterone levels begin to drop. The corpus luteum is not supported anymore. The progesterone and the estradiol cannot support the endometrium, so the endometrium begins to disintegrate and begins to slough off. And so this last 5 or 6 days of the cycle is when we begin to initiate what would be the processes that once we begin sloughing off, that's going to start the new cycle. Okay, so what is our job of all that progesterone while it's high here? The progesterone is going to work at the uterus to mature those cells that the estrogen had just increased the number of. And it's going to decrease the oxytocin receptors because you don't need those small contractions anymore. If the sperm haven't gotten there, too bad, you've got to wait for another try. So cervix, constriction, everything is viscous now, plenty of mucus, and that's what we mean by secretory. The breast has begun to start developing in case you do have a pregnancy. Oviducts, again, decreased, largely more mucus. And you generally have a slight increase in body temperature. So in the olden days, you could tell if you had ovulated by this increase in body temperature. But the thing is, if you're going to use that method, you have to do it first thing in the morning before you get out of bed, because otherwise, once your metabolism rises, as soon as you've gotten up in the morning, you also have an increase in body temperature. So, okay. Now, there are two things I want to cover before we leave. One is what happens when we lose our estrogens. So, so our estrogens are there to inhibit gonadotrophin release, and they're there for two aspects. One is the arcuate nucleus, where we also have opioid peptide in our neurons, and the other one is the preoptic area, where we have the GABAergic in our neurons. So we actually have two different kinds of ways in which the estrogens can inhibit this release. And depending on whether we've got high concentrations or whether it's sustained for days, like it is in the first half of the cycle before luteinizing hormone, or whether we have progesterone around, because if we have progesterone plus the estrogens at high concentrations, then that's going to be inhibiting both FSH and LH release. So, so we've got mechanisms here by which, depending on which hormones we have, we can begin to suppress either one or the other. And we've got two different ways of doing this in the hypothalamus. Now, if pregnancy doesn't occur, then we really do start all over again. When you go home, you can take a look at this diagram, see where these negative and positive feedback are. What is it that they're negative on? What is it that they're positive on? And you can take a look at this. All of those and hormones are all together in this diagram. So you can begin to appreciate the whole complexity of the cycle by looking at this. If pregnancy is initiated, then the corpus luteum is maintained by luteinizing hormone that begins to be suppressed, but human chorionic gonadotrophin is being released instead by the implanted placental tissue. And so you can begin to see this within days after implantation has taken place. And this is where the pregnancy test comes into play, because human chorionic gonadotrophin, while its functioning is very similar to luteinizing hormone, you can make an antibody to it and it won't get confused with or cross-react with luteinizing hormone. So this is the pregnancy test. So the luteinizing is going to be taking place, and then the chorionic gonadotrophin is going to maintain that corpus luteum long enough to make estrogens and progesterones in the first trimester. But in the second and third trimester, then the fetus and the placenta have to take over. And they're taking over by making estriol. So this is a little bit complex, but I think it's important to maybe go over this because the complexity is what's really necessary to maintain pregnancy. And you can tell if a pregnancy is maintained well by the fetal tissue, the placenta, and the mother simply by measuring the amount of estriol that's made. Now the reason is the mother needs estrogens, but the fetus doesn't. So in order to protect the fetus, you can't be making estrogens at the same time. And your androgens are going to protect the fetus from androgenization. And your weak androgens are coming from the fetal adrenal. So we can start out with the mother's blood supply, which doesn't mingle with the fetal blood supply, but the mother can deliver things through the placenta. So the mother is bringing in the nutrients, so the LDL cholesterol can pass into the placenta. The placenta can make pregnenolone, and the pregnenolone can pass into the fetus, where the adrenal can use it to make dihydroepiandrosterone, which you remember is a very weak androgen. And it's even weaker when you put a sulfate on it. So then that can cross back into the placenta, and there's a sulfatase that will take that sulfate out, because sulfates are charged, so you can't get it across until it does. And meanwhile, it gets into the fetal liver, and that can take the sulfate out. So fetal liver, fetal adrenal have to be functioning to get this weak androgen back to the placenta. Now, once it's in the placenta, it can be converted to estradiol and estrone. And then that could cross over. But more importantly, this pathway is taking that dihydroepiandrosterone and converting it finally with aromatase to estriol, and the estriol passes into the mother's circulation. So that's why when you can measure estriol, because it takes a well fetus that has good adrenal and kidney function, and liver function, excuse me, and a placenta that is functioning in order to be able to make the pregnenolone that's necessary here. Keep in mind the progesterone that the placenta makes is now in the second, third trimester, and that's what maintains the progesterone levels in the mother. Okay, so if we don't have our estrogens, we begin to go into menopause, and if we don't have that feedback, then we begin to see very high levels of FSH and LH in the urine. And drug companies can collect up that women's urine and sell it as human menopausal gonadotrophins. So that's where it comes from. And you can visualize what some of the effects of not having estrogen are. These are our typical menopausal symptoms and physical symptoms: atrophy of the vagina and changes in the vaginal secretions and pH, because those were all under estrogen levels. Reduced blood flow because you don't go through those cycles anymore. Sometimes loss of vaginal tone. Osteoporosis, we've already talked about that. So, so I think we've covered quite a bit today. If anybody has any questions, I'll be happy to stay a little bit later and talk about them.